Good day, this is the Professor, CBMD PhD, and today's Wednesday, October 18th, 2023. It is 12:55 and 8 seconds, AM Eastern Standard Time, and today's research, cancer cell atlases. So let's jump to it here. The cell atlases of the human brain, date October 12th, 2023. Source, Karolinska Institute. Researchers have created the most comprehensive atlases of human brain cells to date. The studies provide clues on different brain diseases and give hope for medical advancements in the future, such as new cancer drugs. Knowing what cells constitute a healthy brain and where different cell types are located and how the brain develops from the embryo stage is fundamental to the ability to compare and better understand how diseases arise. There are at present advanced atlases for the mouse brain, but not for the human brain until now. The researchers analyzed more than 3 million individual cell nuclei using the technique of RNA sequencing, which reveals each cell's genetic identity. All in all, the researchers studied cells from just over 100 brain regions and found over 3,000 cell types, some 80% of which were neurons, the remainder being different kinds of glial cells. A lot of researchers focused on the cerebral cortex with the greatest diversity of neurons they found in the brainstem. They think that some of these cells control innate behaviors such as pain reflexes, fear, aggression, and sexuality. The researchers could also see that the cell's identity reflects the place in the brain where they first developed in the fetus, which links to the second project. The developmental cell atlas to analyze over 1 million individual cell nuclei from 27 embryos at different stages of development between 5 and 14 weeks of fertilization. The study enabled the researchers to show how the entire brain develops and is organized over time. Even though the results are examples of molecular biological basic research, the new knowledge generated can also lay the groundwork for medical advancements. The research group has used similar methods to examine different kinds of brain tumors, one of which was a glioblastoma, a cancer with a poor prognosis. The tumor cells resemble immature stem cells, and it looks like they are trying to form a brain, but in a totally disorganized way. What they observed was that these cancer cells activated hundreds of genes that are specific to them, and it might be interesting to dig into whether there are any potential for finding new therapeutic targets. The brain atlases will be freely available to researchers around the world so that they can compare the brain diseases they are researching with what a normally developed brain looks like. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Teacher of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.